I'm Bruce Bugbee, President of Apogee Instruments and Professor of Crop Physiology at Utah State University. Today we're going to talk about new sensors from Apogee, and particularly an extended range quantum sensor. We call that an ePAR sensor. I'm first going to show you the sensors and the model numbers, and then I'm going to go through why Apogee developed these sensors. It comes from new research on the effects of spectra on photosynthesis. So let's take a look at these sensors here. These are extended photon flux density and the new one we call ePAR. Now, these are similar price, but look what happens. Let's take this. Am I on? There we go. Let's put the pen on green here. First of all, photosynthesis, we say it starts at 400, it stops at 700, and it's equal in between. So this area right in here is classic photosynthetic radiation. We've used that for a half a century since the days of Keith McCree as pioneering work. This is called PPFD. I know I just wrote over that, but photosynthetic photon flux density. Okay, and this is the classic Apogee sensor. This sensor is a big seller for Apogee. And this one, let me put it under here. Model 500. That's the classic sensor that measures this radiation that I just drew in here. So why do we have other sensors if that's the classic sensor? There are emerging definitions of photosynthetic radiation that I'm going to talk about. First of all, as most people know, here it is right here, UV radiation right here. This has some photosynthetic effects, not many, but some. I'll talk about them. This radiation out here is far red, after 700. This has significant photosynthetic effects, and I'm going to talk about those in detail in the next few minutes, which is why we've developed sensors to do this. Now, this radiation out here is called extended PFD, and Apogee has sold that sensor now for about a year. It goes all the way out to 1,000 nanometers, and LED lights don't have any radiation out here, so it doesn't measure them. Sunlight has lots of radiation, so you can't use that sensor in sunlight. The breakthrough is Apogee developed a cutoff filter to cut this off right here, about 750 nanometers, so that it now gets, and now we got to show that in red because that's far red, it now gets this amount of radiation in here. This is a big deal because there is a far red LED that many manufacturers, most even, are adding to their lights, and the classic sensor doesn't catch those photons, doesn't show them at all, and they cause photosynthesis. So with this new sensor, which, you know, we're working on catchy titles, so this is extended, lowercase e, extended PAR, model SQ610, that's the one that captures this extra radiation out here. There is another Apogee sensor, I'll show this first, that we call the PAR for photosynthetic radiation right here, and the FAR for the far red radiation right here. This sensor has two detectors in it, and it measures these separately. It measures the classic PPFD, I'm going to write that up here, classic photosynthetic photon flux density, and it also measures the far red. Now this sensor, of course, can add those two numbers together to give you a total, but it does them separately. This is a terrific sensor, way cheaper than a spectral radiometer, and it separately measures these two types of radiation. It is about 25% more money than the other sensors. So save up. You could buy the classic, or right now, we'll go back, you could buy either of these for a little bit more. And they're simple to use, a single number. Now, these are the sensors. What we call an instrument means a sensor connected to a meter. And now I'm going to review the two types of meters that Apogee sells, because it's a package. For many, many years, Apogee has sold this classic meter. There's the on button right there. Couldn't be simpler to use. You grab it, you push on button. This one does record, but it's not very elegant for recording. It's mostly people use it for instantaneous measurements. And MQ means meter quantum. So there's the meter quantum with 610 and the meter quantum with 620. That's the classic package. To make the jump to light speed with this, about a year ago, Apogee introduced a microcache with Bluetooth. So now the same sensors plug into this, and it records data for a long time, over a year. But it also displays the data to your phone with the Apogee app that comes with it. When we go to the lab, I'm old school, right? We're just going to make a quick measurement. I grab this meter because it has one on button. My students laugh at me. They say, get with the program. Just get your phone out and look at all the data you can collect so fast. All of the students, if this meter is sitting on the counter, they say, where's the microcache? That's the one I want to use. That has more data. They can put that in a growth chamber, close the door, and measure what's going on inside. For me, I got to run the cable through the door, and it's a mess. So this is an up-and-coming instrument because of the ability to record. It shows both instantaneous and long-term. There's the picture. I got one here, little microcache. I didn't plan to show it with my phone, but there's some of the histograms. Here's the PAR sensor, and here's the new sensor in a silver housing. You can tell the sensors apart because of their colors, distinctive colors. All right. This is the end of the section on the new sensors. Let's just review this, and now I'm going to go into why it's important to measure these. Nope, I'm going to go back. So just to review, the old classic is extended. It gets this radiation out in here. The improvement to that really is this ePAR sensor, model 610. Then we go to the next one, the PAR sensor, so red and far red separately. If you can afford it, this is a great sensor. But this one doesn't work with a meter, by the way. It's got two outputs, so a single meter. You have to use this with the microcache. And then there's the options for the meters. Okay, now I want to talk about why we want to measure these different types of radiation. And this is a much longer part of this talk, but you can skip around in the video to the parts you want. I like to call this towards an optimal spectral quality for plant growth and development. And this is the name of textbooks, but when we say plant growth, we really mean direct effects on photosynthesis. The dry weight of the plant gets bigger. When we say development, we mean plant shape. The plant, tall, spindly, short, fat, we can manipulate shape with quality of light, and we want to know how the quality of light affects photosynthesis. Plants sometimes get big because we change their leaf shape. If you can make a bigger leaf with light quality, it captures more light, and the photosynthesis per unit leaf area is really the same. So this is a tricky thing, and I keep coming back to this. Plant growth and development, photosynthesis and shape or morphology of the plant. This is very significant because in my laboratory at Utah State University, we have been working on these two parameters full time to get them right. Shuyang Zhen and I have published 2 papers just in the last year on this exact topic, and this is primarily on the effects of wavelengths of light on photosynthesis. They're in these 2 papers. We'll put these in green. They're in Plant Cell and Environment and Frontiers in Plant Science, 2 of our most prestigious high-impact journals. They have long titles. You can pause this and look at the titles, or just search on Zhen and Bugbee, and you'll find the papers will come right up in Google Scholar. Both of them are on effects of photosynthesis. This science is what led to the ePAR sensor. We could see the need for a sensor based on this work. Here's Shuyang Zhen, and I'm very pleased to announce that she is now accepted a position as an assistant professor at Texas A&M University. She's going to be continuing this work there. It's very exciting work because it fundamentally changes the definition of what we call photosynthetic radiation. We couldn't have done this work without funding from both NASA and the USDA, and I want to acknowledge their help. For NASA, we're growing plants on Mars and a colony on Mars, and for USDA, back to Earth. This is the LAMP project, Lighting Approaches to Maximize Profit. So it's a very applied, focused project for greenhouses and growth chambers on Earth. We're very grateful to have this funding to be able to do this work. All right, let's take a closer look at this. If we're going to change the definition of photosynthetic radiation, which has been for years and years 400 to 700 nanometers, we make students memorize those numbers over and over again. Those are the wavelengths of photosynthesis. Now we're going to, I'll put this in yellow, 400 to 750. Another 50 nanometers. I love this quote from Carl Sagan in Cosmos. Extraordinary claims require extraordinary proof. This is an extraordinary claim. We're going to change this definition. The old sensors aren't going to capture that radiation. So here's the extraordinary proof. These are 2 slides that into the weeds on the biochemistry of photosynthesis. This isn't just an experimental measurement. It's supplemented with a lot of detailed theoretical considerations, and I'll walk you through this. This is a gorgeous slide made by my colleague Mark Van Eersel, who is a professor at the University of Georgia. Shuyang Zhen, by the way, got her PhD with Mark working on this stuff, and then she came to my lab as a postdoc, and now she's a professor. This is out of Molecular Cell Biology 6th edition. This is a textbook, but Mark has annotated this with what happens to a photon, and this is a wonderful little animation. Here comes a photon, boom, it hits. It's absorbed. It's a reaction center, this right here, electron. Boom! That excites the electron, moves up to a higher excitation level, then more photons come, hits an absorption site over here, and that then moves that electron, boom, all the way up to there, all the way from the sun to here, to here, to here, to here, out. That's called the light reactions of photosynthesis, and those reactions make adenosine triphosphate and NADPH. That's the light reactions of photosynthesis. And if you can answer that on a test, you're ready to graduate. This is a detailed light reactions. Now, that is a lot of biochemistry, and I really like physics more than chemistry, so I made a different diagram. And you can be the judge of which one's easier to understand. We know from Albert Einstein and the Stark-Einstein law, one photon excites one electron. That's what Mark's diagram just showed us. But now I've got workers on towers more than the biochemistry of this. Here's the sun, right? So we're going to show the sun here, same as the other one. The sun is always smiling. Photons come in, boom! This guy hits this platform with the hammer, the electron goes up here, another worker catches it. Note that there's male workers here and female workers on doing the skilled positions at the top. They put it in here, it turns a mill, and it makes ATP, the energy for photosynthesis. Another photon comes in. This one's coming in this way, boom! Sends it up here, another worker puts it in a bucket, and we get NADPH. This is called photosystem 2 and photosystem 1. We knew what 1 first, that's why they're backwards. 2 happens before 1. But it takes photons from both sides to do this, to fully excite all the electrons for photosynthesis. Now, we have known for over 50 years that this worker right here can be excited with far red photons, above 700 nanometers. But you can see if this guy was the only one that was excited, this system wouldn't work, because you got to have this side. You got to have him going up here, and then he does the second jump. This is synergism, and we've tried with far red light and it does nothing. And we say, well, that's no good. We got to have visible light. When we apply them both at the same time together, this guy is excited, this one is excited, and the whole system is more synergistic. Now, Shuyang worked on this in Georgia and showed that there was less heat produced when she gave plants far red light. When she came to Utah, we started. This is called the Emerson enhancement effect, because it was shown by Emerson. Amazingly enough, in the 1950s is when Emerson, and then he was tragically killed in a crash of a small plane, and no one ever followed up his work. It's an amazing story in science. So Shuyang comes along, and we start doing canopy photosynthesis. Whole canopies of plants in a closed box, you can measure photosynthesis. And we get this classic curve with 400 to 700. This was with lettuce now. And it starts here at 0, goes up like this. And 3 points in this curve. When we gave them far red light, photosynthesis went up, but right here, this is 400 to 700. We weren't counting it, right? You don't count those photons. Whoa, but it made photosynthesis go up. These are different levels, went up. So what Shuyang did, graph this, 400 to 750 nanometers. She counted it, here it is. And look, all the data follows one nice smooth curve because we're counting all the photons. When we got these experimental results, we went, wow, this is beautiful, but so far we've only showed it in lettuce. What about other crops? Shuyang did 17 crops. Spinach, basil, kale, sunflower, corn. You can see them all on here. And this is a picture of the plants in the chambers, one at a time. To make a long story short, every crop did the exact same thing. So this effect of far red photons is universal for many kinds of crops. This led to what I'd like to say is going to become a landmark publication in Plant Cell and Environment from this work. But we kept going. We kept working on this. The second paper, which is now published in Frontiers in Plant Science, looked at what we call quantum yield over the long term. So this was like several weeks of a lettuce crop. And quantum yield is, we have it right here, moles of carbon fixed per mole of photons absorbed. It's a very precise measurement of photosynthetic efficiency. And here's 350. This is micromoles. I'll put it over here. Micromoles per meter squared per second. That's PPFD. Note that this one had 350. This one only had 300. So this one should have been much less. But we added 50 of far red. So the sum of these two is the same, and the quantum yield is the same. Down here, this was with RB is a red-blue background, like purple light, and it was the same with purple light. Then we also did the same thing with white light, 350 and 300 plus 50, and the lines are the same. That's compelling evidence that far-red photons are equally good to traditional photosynthetic photons, and that's the underlying reason we need a sensor that includes them, particularly for LEDs, because all those LEDs have far-red photons now, and we need to measure them. So that ePAR sensor includes these. So there's the classic, there's the new 750. The ePAR sensor, if you look at this, includes some photons here. Those are UV photons. And now I'm going to briefly talk about what UV does, and I'm going to base it both on McCree's data and our data. Remember Keith McCree, who he's a wonderful guy. As a young scientist, I had a chance to meet him, and he said, "Bruce, what are you working on?" And I told him. And first he said, "Bruce, what's the most important question in your discipline?" And I thought, "Oh yeah, it's probably this." And he said, "What are you working on?" And I told him, and he says, "Why aren't you working on the most important thing in your discipline? Get back to the lab, get to work." It was really inspiring. So he gave us this. Now we're adding on this. What about right here? Is this a sharp cutoff right at 400? This is particularly important because now we have LEDs right in this region, right here. Now I'm going to show you these LEDs and show you why this end is also important. Here's 4 colors of LEDs. I'll put this one in blue. This one we call violet. We don't have the names on here, but violet is really quite an efficient LED, and it's right on the edge. Right here, we call it UV, and look at this. A big part of that sensor is really UV radiation. Do these cause photosynthesis? Yeah, let's take a look. Are these efficient over here? This sensor is just inside of UV radiation, but 2.22 micromoles per joule, that's an efficient LED. If this does unique things, we need to be putting this in fixtures. These 3 are primarily UV. This one, though, has a tail in the visible. So this is why we need to know exactly what's going on right here with photosynthesis. Look what happens. 65% efficient, excellent. 58, 57, 49. These become less efficient, but if they have unique effects, we need to be using them. This one is becoming increasingly used. These 3 are still emerging possibilities, but if we can show they help, fixture manufacturers will use them. Here's the same graph, just shown on a bigger scale. This is UVC down here, which we're also studying. This is very damaging, but you just give trace amounts and it has the potential to do good things. So here's the same thing. The peak heights are proportional to their efficiencies or their efficacies. This is the standard blue right here, 450 nanometers. White LEDs start from this. This is very common in all fixtures. Here's the violet right here. We'll put V for violet. And then the three wavelengths of UV. What do they do to plants? Here's McCree's curve that everybody shows this, and we look at it over and over again. What we just talked about, shown in red, is this curve right here. We've now got compelling evidence that we need to shift this. What about this end? Look at the tail on this end and compare it to those three LED curves. What does this look like? Let's take a deeper dive into McCree's original papers on this to look at what the photosynthesis looked like. This is a graph we made from McCree's data, and here is 400 nanometers right there. These are plants from the field, and they sparingly use that UV light. The growth chamber plants, wow, look at this. The growth chamber plants use the UV light. I used to think this was because they weren't used to UV and they didn't filter it out and they used it all the time. But on closer inspection of his data, coupled with our own data of measurements of plants, now we know this is it was just chance. They have used different species here in the growth chamber and the field. It isn't because they were grown in the growth chamber, he just used different species. Let's take a look at this. There's the UV that we're talking about. Look at the big error bars in here. This is a fascinating area of study. Here's an example. Lettuce and cucumber, both two high-value common species. Here's 400 right there. Cucumber minimally uses UV light. Lettuce uses a lot of UV light. It's much more effectively uses those UV photons. Now, I could show you all the other crops we've studied, but sunflowers is one that, sunflowers is actually pretty bad. Sunflowers, you'd think they were good. Some species are even more out here, but there's a big range of curves in the UV among species. So, for this reason, and I'll stop, we got one more summary slide. For this reason, we have made, this is the old data from McCree where it cuts off here. The new sensor comes out here, comes across at 1, all the way across, and then it doesn't have a sharp cutoff. It counts some of these UV photons, has a tapering response there. So the advantage is a little bit of UV right here and a bunch of far red there. That's the ePAR sensor. Now I'm going to close with a slide that summarizes all the types of light on plants. Blue photons inhibit cell expansion. They make plants small and compact. Green photons they're fine for photosynthesis, but they're really important for human vision. If we have green photons, we can see the plants. Red photons are terrific. Efficient photosynthesis. Our fixtures have a lot of red photons. Far red photons do the opposite of blue. This inhibits cell expansion. These promote it. So as we go into the future, we're going to be going back and forth between inhibiting and enhancing cell expansion with these colors of light to shape the plant with photobiology. Here's an example. This happens to be cannabis, 5% blue, 20%. We shrunk the plant. Everything else was identical here. And generally, a nice compact plant is a good thing, but this plant is also a little bit wider than that plant, and it captures more light. As you can see how, now we could put far red, if I do that in red, if we put far red in here, this is going to look just like that, with more blue. It's a fascinating ability to manipulate plant shape. Human vision, you can see the green. This is white LEDs. You can see the plants. There you can't. That's significant. Here's far red light in lettuce. It's tremendously valuable. Look at that. 10% far red leaf expansion. It's really valuable for lettuce. Unfortunately, the far red light makes most plants significantly taller. So we have to be very careful how we use it, because it can easily make, well, tall, skinny plants instead of short, compact plants. This is all in front of us, manipulating far red, putting it on at the right place and the right time and the right crop. But it's a very powerful tool, and that new ePAR sensor now measures it. Thanks for listening. We'll talk to you again in another video.